My name is Maria Fredriksson and I work at Lund University, and I'm going to talk about tuning wood-water interactions by targeted wood modification. And first I would like to say that this is a product of teamwork, so I would like also to acknowledge my excellent colleagues, Ramona Stigitis, Emil Tybring and Lisbeth Tygesen, who also provided a lot of input to this work. So, as you may know, if we place wood or wood-based materials in moist or wet environments, it will eventually be degraded by fungi. And one essential requirement for fungal degradation to occur is that water is present. And if we look in literature, we can see that the minimum water potentials for fungal degradation ranges between -4 to -0.1 megapascals. And if we translate that to relative humidity, it corresponds to levels between 97 and 99.9% relative humidity. So we can conclude that it's quite high moisture levels that are needed in order for fungal degradation to occur. And what do we know more? We know that it may not be only the amount of water that is important for fungal degradation. It might be so that also the location of water within the wood structure is important. Because the fungi needs water for different purposes. We have known for quite some time that fungal hyphae need capillary water in cell lumina, because that water is accessible for the fungal hyphae. But more recent research has also put focus on the moisture content of cell walls. It could be so that a minimum moisture content in cell walls is needed in order for a transport of the substances that are secreted by the fungi. Because if the moisture content in the cell wall is too low, the pathways for transport could be too narrow or non-existent, and that would not make it possible for these substances to be transported into the cell wall, and thus the wood cannot be degraded. So one reasonable strategy to prevent degradation of wood would therefore be to limit the access of water. And there are such modifications, several different types, and one of these is modification by acetic anhydride, which is also called acetylation. And this modification is known to significantly increase the resistance to degradation. And we know that acetylation decreases the cell wall moisture content, which could be a mechanism, but we also know that acetylation changes the interaction between wood and the water in cell lumina. But we don't know what the exact mechanisms are that makes this such an effective measure to prevent the wood from degradation. So in order to reveal that, we would need to do experiments where we can limit the fungi's access to water in different parts of the wood structure separately. So the question we asked ourselves before starting this work was if it's possible to change the interaction with water and wood differently in different parts of the wood structure. Because if we could do that, we could also investigate the influence of access in water in cell walls separately from access to water in cell lumina, for example. So what we tried was to take spruce as a material, and since we know that modification with acetic anhydride is known to increase the resistance to decay, and it's quite straightforward to do modification with acetic anhydride, we chose that for our first experiments. But we wanted to have two different modification strategies. And one of those was to modify only the interface between the cell lumina and the cell wall. I call it that the interface modification here. And one was to uniformly modify the whole cell wall. And the point of this was that our theory was that interface modification then would not significantly affect the interaction between the cell wall water and the wood, but influence the interaction between the water in cell lumina and the wood, while the uniform modification most probably would influence both types of water. So we used acetic anhydride, and we used different temperatures and reaction times to achieve different degrees of modification. And in the case of uniform modification, we used pyridine as a swelling agent to enable a good penetration of the acetic anhydride into the cell wall. So then, of course, we wanted to know, did these modifications work as we hoped them to work? So then we use Raman microspectroscopy imaging to figure that out. And here are some results from those measurements. What we see in this picture is the peak that represents the C-double-O bond, which is seen in the acetylated parts of the wood. So the more yellow the picture are in these images, the higher degree of modification we have in the wood. So we can see the untreated spruce here up as a reference, and we can see that for the interface modification, which we did to do 2 different degrees, we can see a light modification at the surface in case A, and a more higher intensity modification in picture 2. But in both cases, the modification only acted at the surface, cell wall surface, not through the cell wall. While if we take a look at the uniform modification, which we did to 3 different degrees using 3 different reaction times, we can also see that we managed to do this modification so that it was distributed uniformly in the cell wall. And we can see by increased the reaction time, we also get an increased degree of modification from A to C. So after doing this, we concluded that it seemed to be possible to control how much of the cell wall that was modified. And then we wanted to continue, of course, to our main question: does this change the interactions between wood and water? And in that case, how? So to study that, we use a technique called low-field NMR, which can be used to separate different water populations within the wood structure. And to get a sense of what it looks like, we can first take a look here on untreated, water-saturated spruce samples. So this is what you get if you do low-field NMR measurement on a saturated softwood. If you have hardwood, you get more peaks, since hardwoods include more different types of cells. But we can see here that we have 3 distinct peaks, and they represent, from the left, water in cell walls, water in small macrovoids, and that is, for example, pit chambers, and the large peak to the right represent water in cell lumina. And you get this separation since the relaxation time, which we have on the x-axis, T2, is influenced by physical environment, that is essentially pore size, and therefore we get this distinct separation of the water in different locations. We should also remember, though, that T2 is not only influenced by physical environment, it's also influenced by chemical environment. So what did this look like for our modified samples? So if we first take a look at the uniformly modified samples, it looks like this. We can see that all peaks shift to the right. And a shift to the right, if that was due to the physical environment, would mean that the pores are larger. And we know that this type of modification does not suddenly increase the size of the cell lumina. So the shift of the peak here is rather due to a change in chemical environment than a change in physical environment. So we can see here that for all 3 peaks, we see a peak shift to the right, which indicates a change in chemical environment, which we also expected. But if we take a look at the interface modification results, first we can take a look at the higher degree of uniform modification, and we can see that the higher degree of modification, that is longer reaction time, the more these peaks shift to the right, which was also quite what one could expect. But now we can take a look at the results from the interface modification. And there we can see for the large peak to the right, representing cell lumen water, we can see that there is actually a shift, a smaller shift for the lower intensity interface modification, and a larger shift for the higher intensity interface modification. But if we take a look at the cell wall water peak, the change is much smaller, or even no change for the lower intensity interface modification. So it seems as if we're using this modification, we have managed to change the interaction between water in cell lumina and wood without significantly changing the interaction between water in cell walls and wood. So all these measurements were done on water saturated samples, but then it would be nice to see what it looks for other high moisture levels. And as you might remember from the beginning, we concluded that if we are going to keep in the range which is relevant for fungal degradation, it's the high moisture range that is relevant, 98–99% relative humidity and above. And therefore, if you want to create well-defined moisture states and measure the distribution of water within the wood, you cannot use the techniques that you normally use for lower moisture levels. For lower moisture levels, you can use condition samples above, for example, saturated salt solutions. But that's not possible when we come to the range which is relevant for fungal degradation. Then we need to use a technique called the pressure plate technique. And I will not go into detail on this now. You can read this paper if you want more information. But it's basically a technique where you can get well-defined high moisture levels. So just to show you some examples, these data are quite new and they are not fully evaluated, but these are some examples where you can see how the distribution of water within the wood changes for the different treatments and at different high moisture levels. And using this equipment, we can do that both in desorption and also in absorption. So we can see in absorption at the same moisture level, there is no water in cell lumina, for example, while there is still water in cell lumina in desorption. So to conclude, from this study, it was possible to alter the interaction between water differently in different parts of the wood structure. And we thought also this methodology with combining low field NMR and pressure plate conditioning is promising in terms of monitoring water distributions within the wood structure. And we think that this would be a tool to do systematic studies on the role of water in fungal degradation processes. So if you have questions or comments or ideas on how to further develop this, you're welcome to contact me or ask questions now or contact me at a later point. Thank you very much.